Okay, guys, today I will teach you how to create a phylogenetic tree. So, this is the software we will use, the Mega. It's one of the most commonly used software for simple phylogenetic analysis. Um, and the tree making is one of the most fundamental aspects of molecular evolution and molecular genetic studies. So, before making a tree, you have to have a protein sequences. Here we have protein sequences. I have selected certain protein sequences. Um, Whenever you are going to use protein sequences, you should keep in mind that your data is very simplified. It's in FASTA format, and the headers, the titles of the sequences are very simple, and there is no gap between different part of the sequence titles. For example, kindling is the protein name, and then Homo sapien is the species, and it is written very simplified in a very simplified way, and there is no gap whatsoever in the different parts of the title. So I copy this sequence now, and I put it into the Mega. I go to the align. This is Mega 7 software. There is also Mega 10 and other versions, advanced versions. I mean modern versions, but I prefer Mega 7 because of its very nice looking interface. It's very, I would say, simple, very easy to use. So, added alignment, create a new alignment. It's protein sequence, of course. We paste sequence, fine, and we make for alignment. We go for the cluster W. We use the default parameters, and fine, it's fine. Now we go to data, and we use the data for phylogenetic analysis. Now we come to the main software interface again, and we go to the phylogeny, and in the phylogeny we use the neighbor joining tree. You can also generate maximum likelihood tree or whatever tree you like, but for the sake of convenience I am making now the neighbor joining tree because it's the easiest of the tree and it's very simple tree. So yes, we want to use this data, and I'm using this default parameters. One thing is the bootstrap method is a method for the validation of your process. So I use you is at least 1,000 uh, value should be used for the bootstrapping for the sake of convenience because we don't want to spend much time. I am just taking to 10, but you should always use it 1,000. And the other things I keep it like that, and we compute it. And after computing, so we have here our tree is made. Now this is our tree here. You can see it in this form. This is one of the form in which you can also you can see the variation in the length of the branches, which let you know about the distances between different sequences. But you can also produce some other, let's that you can use these straight ones. You can use these ones. You can also use toggle it so that you can actually see the distances between different sequences. So this is about making the phylogenetic tree. But what is a phylogenetic tree? What does it tell you? I will let you know about it in a separate episode. Thank you.